Hello viewers, today I'll be talking about the life cycle of the most two common bryophytes found universally. I'll be talking about Mercantia belonging to the order Hepaticopsida and Funaria belonging to the order Bryopsida. I'll be talking about their morphology, their anatomical features and how do they complete their life cycle. So basically let's start with the simplest bryophyte, that is Mercantia. Mercantia, the genus, has got 65 species distributed worldwide. It grows best in cool, moist and shady places. The most common species of Mercantia is Mercantia polymorpha. Generally, there are found to be 11 species of Mercantia growing in India, mainly in the western Himalayas. Let's move forward towards the structure or the morphology of Mercantia. Mercantia, basically like the other bryophytes, has got a very simple gametophytic structure. It starts the life cycle with a simple gametophyte structure that is in the form of thallus. The thallus is a dorsi-ventrally flattened structure with no differentiation into stem and leaves. The thallus organization of Mercantia shows it to be a dorsi-ventrally flattened and prostate structure. As you can see from this diagram, it is a dorsi-ventrally flattened structure with the thallus at its deeply notched. Along the midrib, there are numerous gamey cups. On the ventral surface of the thallus, as you can see, it is much more lighter in color than the dorsal surface and it contains numerous rhizoids. Rhizoids, these are the unicellular structures and they develop from the ventral epidermal cells. The rhizoids are unicellular, unbranched structures which helps in absorption of water and minerals from the soil. It also helps in anchoring the thallus body into the soil. The rhizoids are of two main types. These are basically the smooth rhizoids and the tuberculate rhizoids. Moving forward ahead, so let's see the anatomical, or before proceeding to anatomical, there are a number of distinct structures present on the dorsal surface of thallus. The dorsal surface of the thallus can be divided into a large number of regular, rhomboidal or polygonal areas called areolae. Each of the areolae has got a distinct air pore and the air pore is surrounded by 4 to 8 superimposed tiers of cells. Now moving forward into the anatomical features, the Mercantia, if we cut the transverse section of thallus of Mercantia, shows three distinct regions. That is, it has an epidermal region. The epidermal region are of two types, the upper epidermis, the lower epidermis, followed by a photosynthetic region and a storage region. The epidermis is made from the epidermal cell. The epidermis is not continuous, it is interrupted by the air pores. Below the upper epidermal layer is the photosynthetic region. The photosynthetic region consists of air chambers, or it is composed or it is demarcated into groups and these tiers or the groups are coming or they are made from photosynthetic filaments. From the base of the photosynthetic region are the photosynthetic filaments. The photosynthetic regions are demarcated by tiers of single cells. The photosynthetic region or below the photosynthetic region is the storage region. The storage region consists of isodiametrically placed parenchymatous cells and they store starch in it. Below the storage region is the ventral epidermal cell or the lower epidermal cell, and from the lower epidermis arises the rhizoids. So this goes for the anatomical features of Mercantia. Mercantia reproduces, or the gametophytic structure reproduces by means of gamey cups. The gamey cups, they occur along the midrib of Mercantia. The gamey, if you look into the structure of a gamey, so it is a stalked structure. It has a one cell stalk and a multicellular, vertically oriented, bilateral, symmetrical discoid body. The mucilage hairs present in the gamete cups helps to secrete mucilage, and this mucilage absorbs water and helps in the dispersal of gamete cups or gamete. From the gamete arises another gametophytic structure. Now moving ahead to the reproduction, the kind of reproduction is the sexual reproduction in Mertensia. So, the sexual reproduction in Mertensia takes place when there are the long days, humid conditions in the soil. Mertensia is dioecious as the male and female organs are called antheridia and archegonia. They respectively occur on different thalli. The antheridia and archegonia are borne on special erect and stalked structures called gametophores. The antheridia or the gametophore carrying the antheridia is known as antheridiophore. Archegonia is known as archegoniophore. So, this is how it looks like. If you look into the antheridia, it is the gametophore, they have the same kind of anatomical features similar to that of the gametophyte. So, the presence of the vegetative tissue is same in the gametophyte. The antheridiophores in case of Mertensia are terminal in position on the thallus. Each antheridiophore consists of erect long stalk and bearing at its top a lobed disc called a receptacle. The receptacle is a lobed structure. Usually a single short stalk antheridium is present inside each antheridial chamber. The antheridia are present on this receptacle in acropetal succession. Acropetal means the younger ones are towards the apex while the matured antheridia occurs in the center position. Now, the structure of antheridium, each antheridium, if you see, it is a club-shaped structure. So, it is a short globular body attached to the floor of the chamber by a short multicellular stalk. The globular body contains a mass of androcytes or the androcytes mother cells which remain enclosed in a single jacket. These androcytes, they transform into antherozoites. The cells of the jacket are thin-walled and these are sterile in nature. Each antherozoid is a small, coiled, uninucleate and biflagellate structure with one flagella beating forward, another flagella beating backward. Archegoniophore is the gametophore carrying the archegonia. So, the archegoniophore, as we can see in the diagram, it consists of a stalk and a lobed disc. After the formation of rosette-like 8-lobed disc, the archegonia appears on the dorsal surface in acropetal succession on each lobe. This downward growth is followed by the inward curvature of the lobes, thus shifting the archegonia from dorsal to ventral surface with their neck inverted downwards. After the inversion of archegonia, one layered plate of tissue develops from each side of the archegonia and this is known as pericytium or involucre. A mature archegonium of Mertensia appears as flower-shaped structure raised on a short multicellular stalk and consists of a swollen base called venter and it has a long slender tubular neck. The venter is surrounded by one thick wall and it encloses a large egg and a small ventral canal cell. The neck of archegonium is composed of vertical rows of cells and at its top are present 4 cover cells which close the upper end. During fertilization or when there is maturation of archegonium, so the archegonium matures and as the egg matures, the ventral canal cell and the neck canal cells separate from the wall of the archegonium and undergo degeneration. The disintegrated material absorbs water and swells up. The swelling force opens the cover cells and a small amount of mucilage pushes open the cover cells and the archegonium is ready for fertilization. The antherozoites, after they become matured, they reach the archegonium and then down through the neck they enter the ovum and then they go for fertilization. So, this diagram, as you can see here, it illustrates the various stages in fertilization or during fertilization. So, here is the archegonium. The archegonium is inverted in structure, which is containing the egg present in the venter of the archegonium. The sperms which are dispersed by the rain enters it, and there it forms the embryo. From the embryo, so the embryo is now becoming 2N. From the embryo, the sporophytic phase of the bryophytes initiates, or the sporophytic phase of the Mercantia initiates. So, this is now 2N. So, sporophytic structure is diploid in nature, whereas the gametophyte is haploid in nature. So, let us move towards the sporophytic structure. The sporophyte is a very short-lived structure of Mercantia and is dependent on gametophyte for nutrition. So, this is the sporophyte, as you can see the structure. So, this is dependent on the gametophyte for nutrition. So, in case of the Mercantia, the gametophyte phase is the most prominent phase occurring in the bryophytes. The sporophytic structure can be divided into 3 distinct regions. The 3 distinct regions are foot, seta and the capsule. Now, let us come to the individual functions of these 3 regions. Foot helps in anchorage and absorbs all the nutrients from the gametophyte or from the developing sporophyte. Seta, which is short and a stout structure and consists of parenchymatous cells. With maturation of sporophyte, the seta undergoes repeated divisions, transverse divisions followed by elongation. Hence, the seta increases in length. When the seta increases in length, it ruptures the calyptra and the pericytium, which is surrounding the developing zygote. So, hence the seta ruptures the calyptra and pushes the capsule out through the perigynium and pericytium. Let us move to the capsule. So, this is the oval shaped structure present at the end of the seta is the capsule. Capsule is a yellow oval shaped structure present at the top of seta. It encloses spores and elaters. The spores are small and round. The elaters are long, narrow, tapering at both the ends. So, as a result of their coiling and uncoiling or due to variations in moisture, the spores are separated and dispersed from the capsule. So, who helps in the dispersal? It is the elaters. This diagram beautifully represents the life cycle of Mercantia. As I said, in the life cycle of Mercantia, it shows alternation of generations. The gametophytic structure alternates with the sporophytic structure. The gametophyte is haploid in nature, whereas the sporophyte is diploid in nature. The gametophyte structure is containing a thallus. The thallus is a very simple thallus in the case of Mercantia. It is not differentiated into the rhizoid, stems and scales. It only contains a simple structure which is known as the thallus, which is dorsiventrally flattened prostrate structure, and it contains at its apex a notch. It branches by dichotomous branching. So, this is how the thallus of Mercantia looks like. Along the thallus, as you can see, there are gamma cups. This gamma cups contains gamma, which helps in the vegetative reproduction of Mercantia. So, this is the most prominent gametophytic phase of Mercantia. Now, the sexual reproduction, as you can see, it starts with the production of gametophore. Gametophore are long stout structures and their anatomical features are very much similar to the vegetative features of Mercantia. The archegonia are born at the top of this gametophore, antheridia are born at the top of this gametophore. The gametophore containing the antheridia is antheridiophore, the gametophore containing archegonium is the archegoniophore. Now, as you can see, Antheridium is a lobe structure, whereas the Archegoniophore is arranged in the form of rosette-like structure. Coming into the details, as I have already discussed, so this is the antheridia. The vertical longitudinal section of an Antheridiophore shows the arrangement of antheridia. The antheridium are arranged in a acropictal succession on the receptacle. Whereas, as you can see, each antheridium alternates with an air chamber. The Antheridiophore, it contains, or the antheridium contains the androcyte producing cells, and these androcytes on maturation produces this kind of a biflagellated sperm. Whereas the Archegoniophore contains the female gametophyte Archegonium. During maturation, the Archegonium becomes inverted in structure. The sperms enters the Archegonium, undergoes fertilization, and this is here how the zygote is formed. The zygote is diploid in nature. From the zygote, the embryo initiates. What is an embryo? Embryo is when the zygote undergoes repeated cell division to form a multicellular structure known as embryo. And from the embryo, we get the sporophytic structure. So, as you can see, the developing embryo is very much dependent on the gametophyte for its food and nutrition. The sporophyte, the details of its structure, the sporophyte contains a foot, a seta and a capsule. The foot helps in anchorage of the sporophyte, the seta, it helps in elongation of the capsule, and the capsule contains pores and elaters. Now, in this capsule, meiosis occurs and the diploid sporophytes, they return to the haploid phase, that is the end phase. And from the meiosis or during meiosis, the spores are produced which are dispersed with the help of elaters, and these spores again produces the protonema. From the protonema, the gametophyte cycle initiates. So, as I said, the gametophyte alternates with the sporophyte. The alternation of generation is due to fertilization and meiosis. Since the alternating phases in this life cycle are dissimilar, so the alternation of generation is of heterologous type. With this, we complete the life cycle of Mercantia. Now we proceed towards the life cycle of a higher bryophyte or a more advanced bryophyte, that is Funaria. So let's start with Funaria. Funaria, as we can see, it belongs to the order Funarilis, family Funariaceae. Now, Funaria is another most common moss growing in the temperate as well as in the tropical regions of the world. In India, it has been recorded that there are 15 species of Funaria. The most common species of Funaria is Funaria hygrometrica. It usually grows on damp soils, moist rocks, and prefers an alkaline environment in the soil. So, as we can see in this diagram that the gametophyte is differentiated into leaf, stem and rhizoids. So, it is more advanced and more evolved than Mercantia. The axis is erect and branched. The branches arise on the main axis from extra-axillary position. Rhizoids, these are the rhizoids, they are slender, branched and multicellular. They help to absorb the water and minerals from the soil and give it to the plant, to the growing gametophyte. The leaves are spirally arranged on the main axis. The lower leaves are small and scattered, while the upper leaves are large and crowded. The leaves are simple, flat, bright green in nature. Anatomy. Moving ahead into the anatomical features of the gametophyte. So, it is well differentiated into an epidermis, cortex and central cylinder. Epidermis, it may be one cell layered or may be two layered at some places. Cortex, it is a multilayered zone and made from the parenchymatous cells. It has a central cylinder. It forms the central core of the stem. It is made from the narrow, thin-walled, compactly arranged cells which are often colorless and lack protoplasm. It imparts mechanical strength to the erect axis and it helps in the conduction of water and solutes. So here we see the development of a vascular cylinder or the vascular zone. The Funaria reproduce through different means of vegetative reproduction. One of the most common means of vegetative reproduction is the production of protonema. The protonema, which develops from the rhizoids, they help in the generation of new gametophytic structures of Funaria. Now let's come to the reproduction, another type, that is the sexual reproduction. So it is basically monoecious in Funaria, that is Antheridia and Archegonia are born on the same plant, but they arise on different branches on the same plant. So the Antheridia are born on the main axis, while the female branch develops as a later branch from below the base of the male branch. The main axis of the gametophore bears Antheridia at its tip. It is easily recognized due to the presence of small and scattered leaves on its lower part and a large number of closely arranged perichetial leaves in the form of rosette, forming a perigonium or perichetial cup at the top. The Antheridia are present within the cup, are present in different developmental stages. Perichetial leaves provide protection to the Antheridia. There are a large number of green, sterile, hair-like filamentous structures called paraphysis, and these paraphysis is multicellular 4 to 6 cell filament, and the terminal cell is a large subspherical and capitate. They provide protection and helps in conserving moisture. Archegonium again is born on branches which are known as archegoniophore, and these arise from the base of the male shoot. The archegoniophore or the archegonia intermingled with paraphysis and are present terminally in groups. Each archegonium is situated on a long and massive multicellular stalk. Each archegonium is flask-shaped and is differentiated into a long neck and a swollen venter. The wall of the archegonium is single layered in the neck region and 2 layered thick in the venter region. It consists of 6 vertical rows of cells in the neck, and the ventral region consists of a neck canal cell and an egg. Now the venter region, as you can see, when it matures, the ventral canal cell and the neck canal cell disintegrate and they form a mucilaginous substance. The mucilaginous substance pushes open the cover cell, and in this way the archegonium becomes ready for fertilization. The fertilization, the similar process where the spermatozoid reaches the ovum and it fertilizes it, and from there the zygote is formed. From the zygote, the multicellular embryo structure develops, and the embryo structure differentiates later with the help of cell division into a sporophytic structure. So the sporophytes becomes or matures when it is still attached to the gametophyte. It is a complex and highly differentiated structure into foot, seta and capsule. Foot is a poorly developed structure of the sporophyte, and it is present within the gametophyte in the female gametophytic shoots, and it functions as an absorbing and anchorage organ. It absorbs water and minerals from the gametophyte. The seta is a long and slender structure. The seta is much more larger in length or longer in length than the seta present in Mercantia. It is differentiated into a central strand surrounded by a cortex and an epidermis. The central strand continues upwards into the tapered base of the capsule. It helps in conduction of water. Capsule, it is situated at the tip of long seta. It is erect or pendant, pear-shaped, green or yellow in color. With maturation, moreover, it turns yellow. It is concerned with the formation and dispersal of the spores. So the capsule in case of Funaria is much more developed. It is again differentiated into 3 parts: apophysis, theca and operculum. Apophysis, it is the basal part of the capsule and appears as an extended upper part of the seta. It contains conducting tissue similar to spongy tissue of seta. Theca, next to apophysis, is the central fertile region called theca. It contains sterile capsular region and fertile sporogenous tissue. The central capsular region is known as the columella. The columella is cone-shaped and it extends upwards into the operculum, and in its narrow part is connected with the central part of the apophysis by a few filaments. Whereas the fertile tissue, that is the sporogenous region, lies in the form of a spore sac and is bounded by an outer spore sac and an inner side by an inner spore sac. So, these spores or the spore sac contain spores which undergoes meiosis or cells which undergoes meiosis and produces the mature spore. Operculum, this is the upper part of the capsule and which encloses peristome. The number of peristome teeth found in Funaria capsule is 32 in 2 whorls. The peristome teeth, what are they? They are teeth-like projections that are present around capsule in a bryophyte to protect it. So, with this we come to the life cycle of Funaria. As you can see, the life cycle of Funaria, typical of the bryophytes, has a dominant gametophytic phase. The gametophytic phase is much more differentiated, and there you get the stem, leaf and rhizoids. From the gametophyte, there arises a female branch and a male branch. The female branch bears the archegonium. The archegonium contains the matured ovum, whereas the male branch contains the antheridium. The antheridium produces the antherozoid, and this antherozoid fuses with the ovum to form the zygote. From the zygote, the embryo is formed, and the embryo is now 2n in number. The embryo produces the sporophyte. The sporophyte in case of Funaria is much more developed and differentiated. It undergoes, the sporophyte contains the spore sac. The spore sac produces or undergoes meiosis to form the spore tetrads. The spore tetrads then becomes dispersed, and these on germination forms the protonema. The protonema then give again rises to the gametophytic structure. So, similar to Mercantia, in Funaria there is alternation of generations. The gametophytes phase, the gametophytic phase alternates with the sporophytic phase due to fertilization and meiosis. The two phases contains two distinct type of morphology, hence there is heterologous life cycle. With this we complete the life cycle of Funaria. So, let us now conclude. What are the common points between Funaria and Mercantia? In both the cases, we saw that the life cycle starts with a gametophytic phase. In case of Mercantia, the gametophyte is a simple thallus, whereas in case of Funaria, it is a bit developed, that it is differentiated into rhizoid, stem and capsule. Both contains the male and the female organs on the tip of the branches. In case of the Funaria or in case of Mercantia, we are calling it as antheridiophore or archegoniophore, whereas in case of Funaria, it is born at the tips of the branches. In both the cases, the life cycle shows heterologous alternation of generations. So, thus the Mercantia is less developed in comparison to Funaria, which is more developed during the course of evolution. Thank you.